Hello, AP Environmental Science students. I'm Dr. Sowell, and we are going to proceed with our review of our content for the exam. I teach APES down here in Jacksonville, Florida. It's hot and swampy and wet today down here. So just as a reminder, here's our exam date, May 18th, and those are the times. And then we've also got the details of what's on the exam, the two FRQs for you to take a look at. So yesterday, Ms. Fassler talked to you about biodiversity and really got into the theory of island biogeography. So just to review some of those concepts on biodiversity, she left you with an FRQ, and we are going to go over that together now. So the first part of your homework asked you to look at this diagram of two forest communities, Community A and Community B, and it says, identify two different scales on which biodiversity can be measured. So it's asking for two different things. So it's 2 points here, and you could have said either genetics, species, habitat, or ecosystem biodiversity, those different levels of biodiversity. So a statement such as biodiversity can be measured by genetic diversity and species diversity would have gotten you both points because it's just an identify, not a describe or explain. The second part says use the diagram to describe one difference in biodiversity between community A and community B. Well, if I just look at these two communities, I see that there are 5 different species of trees here and 5 different species of trees here. So the species richness itself isn't that different, the number of different species, but their abundance is. And so the correct answer would be something like community B, here on the right, is more diverse than community A because B has higher species evenness than A. Or you could have said it this way, community A is less diverse because one species of tree dominates the community. The tree populations in community B, however, are more equal in terms of proportion. So that's just another way to describe the biodiversity between A and B, other than they have the same amount of species. The next part, C, says describe one regulating service provided by a community like the one shown above. So you could have said any kind of regulating service provided by a forest, regulating climate, regulating local air quality by removing pollutants or increasing the amount of shade, storing carbon, sequestering carbon through photosynthesis, moderating extreme weather events, filter water, and there's probably a few more that aren't listed here, but any one of those regulating services for the forest ecosystem, not of a tree, but of a forest. And the last one says, an ecologist is interested in the effect that fire intensity has on the biodiversity of tree communities. Describe the control group for an experiment that examines the effect of fire intensity on the biodiversity of tree communities. So if we're looking for a control group and we're studying fire intensity, well, clearly the control group is the one with no fire or no fire intensity. And that fire intensity of 0 can be used as a comparison for the baseline amount of biodiversity. And this fire is an example of a disturbance that we'll look at today. So that is worth 1 point. Oh, there is one more point. So it also says the diagram illustrates some of the results of the experiment. The experimental group with the highest intensity fire after 20 years is shown in A. The experimental group with the lowest intensity fire after 20 years is shown in B. Based on this information, state the likely original hypothesis. So this is 1 point. So it could have been something like high intensity fires decrease the biodiversity of tree communities or a forest ecosystem. Or you could have said the opposite, low intensity fires increase those things. So we're looking for a hypothesis based on the experiment that is described. So remember, experimental design is part of your FRQ1 in some flavor. All right, so where are we going today? We're going to talk about 3 things, 2.5, 2.6, and 2.7. So Unit 2, topics 5, 6, and 7. Natural disruptions, adaptations, and ecological succession. And a few times along the way, we will stop and look at practice 5 about interpreting quantitative data. All right, so let's pull back a little bit and look at our big idea. So one of the big ideas for our course is this. The Earth is one interconnected system. Natural systems change over time and space. Biogeochemical systems vary in ability to recover from disturbances. So you'll notice that change and response to change, recovery, lack of recovery, really does highlight the fact that ecosystems have structure and diversity that change over time. Our world is not a static place. There's earth processes and systems that cause changes naturally, and organisms must adapt to them. And if they don't, they go extinct. So by the end of this first section on natural disruptions, you should be able to explain how natural disruptions, both short and long term, impact an ecosystem. And this actually will carry through the entire lesson today. But initially, we will define what a disruption is in terms of its characteristics and some of its consequences. We'll look at some examples of the natural disruptions at different scales. And then we'll look at how organisms respond to those disruptions. But again, first, we got to define what we're talking about. A disruption is an event. It's an event that changes the structure and function of an ecosystem. And again, we're looking at natural disruptions here. So here are some examples: a hurricane, a forest fire, a volcanic eruption, a flood, a tsunami, a drought, a wave of insect pests. Any of these events can affect the structure and function of the ecosystem in which they happen. What we're not talking about today are anthropogenic disturbances, human-caused disturbances like tilling a field or clear-cutting a forest, burning fossil fuels to cause climate change, or maybe even fishing, like trawling the ocean floor. So today we're not going to focus too heavily on anthropogenic. We may a little bit, but for the most part, we're looking at what are the natural disruptions that just happen short-term and long-term to our Earth processes and systems and environments that cause organisms to have to respond to them. That's what we're looking at today. All right, so some natural disturbances can have greater impact than some anthropogenic ones. So meteor hitting the planet, causing a big loss of species like the dinosaurs. That's a big impact. That's a lot bigger impact than me clear cutting a forest down the road. But either way, they can really have big effects on habitats. They can wipe out a habitat or fragment it or change the environment. Another example of a large-scale natural disturbance would be sea level rise over time. So if we look over hundreds of millions of years ago, we've seen sea level change rise and fall over time. Well, if you're an organism that lives in a coastal area and sea level rise happens, that's going to force you to either adapt or go extinct. So these natural changes do shape the organisms that live on the planet. And they can occur at different time frames. So our Earth systems don't all happen every Tuesday at 3 o'clock. There's a earthquake kind of a thing. There are certain things that occur on a regular frequency and some things that are more random. So if we think about a periodic change, they occur at pretty much regular frequencies, like the rainy season or a dry season. That's an annual event. The frequency is pretty regular. Then we could have something that's episodic that happens occasionally, but not with a regular frequency, like a hurricane. Here in Florida, we get lots of hurricanes. Some years we have more than others, but there's no particular pattern in number and timing of them that is a regular frequency. And then we have things that are more random, no particular frequency. Now, random doesn't mean that we don't know how they occur. We know what causes a volcano, but the eruption of that volcano or the earthquake or some of those processes don't happen on a particular regular pattern. So that's why we consider it random. All right, so let's look back at Africa. So the other day when we were reviewing terrestrial biomes, we used the HHMI Biointeractive Biome Viewer to pull up some climatograms of different locations. So let's just go back and look at the one here in Mozambique, in Gorongosa in Mozambique. If we just look at temperature and precipitation, we clearly see that during the summer months, the temperature goes down and the precipitation goes down. So we clearly have this disturbance that happens in the summer months here in the southern hemisphere that organisms have to deal with. So this is a distinct seasonal change in precipitation and temperature. It's a periodic disturbance of this dry season, and it affects the structure and population of the organisms that are there. So here's a little clip of that information, especially looking at precipitation. So if you'll notice in the summer months, it starts to dip down and come back up, and you'll see that same pattern in the video here. And all I'm doing is going onto Biome Viewer under precipitation, picking that location, and then changing the months so that you can see that the rainy season and the dry season as a pattern here. So now we're in those summer months, and it's getting back into November, December, and then the rains return. So it is a cyclical pattern there. So how might this periodic natural disruption affect the organisms that live there? Let's take a look at the wildebeest, for example. So the wildebeest migrates in response to those rains. So the seasonal rains are periodic disturbances that shape the behavior of the organisms living on the savanna. So these wildebeest migrate, and not just a couple blocks down the street. This is hundreds and hundreds of miles. This is a big, big migration that happens annually in response to that natural disturbance. This is just a map of a herd moving from in between Tanzania and Kenya around Serengeti National Park. But you can see that there is this annual migration that really does follow those patterns of rain. So obviously, these organisms, these wildebeest, have evolved particular adaptations that benefit this lifestyle of migration. So what are some specific adaptations? So they have a body structure for long travel. So their legs and their musculature really do benefit these long distances they have to travel. Herd movement, they don't do this alone. Safety in numbers, you know, one guy's looking out over here while the other guy's eating. So lots of eyes out for predators. That herd movement is an evolutionary adaptation that's beneficial for these long migrations. They have a particular mouth and lips for grazing on these short grasses that they're following throughout their migration. And finally, another one is calves. So as soon as a young wildebeest is born, within a couple of hours it can stand, and within a couple of days, it can keep up with the herd. That would be an important adaptation for the success of a migration. So how do these adaptations get there in the first place? That's the topic 6 for Unit 2, describing how organisms adapt to their environment. Now, clearly we're talking about evolution here. Now, for the AP Environmental Science course, you need to understand the basic mechanisms of evolution, but we don't go into it as much detail as, say, AP Bio would. So to get to this ability, we are going to explain the connection between these environmental changes, like these disruptions or disturbances, on organism survival. And then we're going to explain in general, very briefly, the mechanisms of that adaptation. So going back to our big idea, remember, change is constant. Change is always happening, and that these disruptions and disturbances are normal. We have natural disruptions and disturbances that occur. And then we could say that they're seasonal, short-term things like the rainfall disturbance, or these big long-term disruptions like sea level rise. But again, like I've said before, organisms must, over time, adapt to the living with these changes, or see you later. So clearly this is coming on a genetic level. So we're looking at DNA here. That is the basis for these adaptations. And so we have our DNA, we're all familiar with that. And then every once in a while, there's a random mutation. And again, that is a random event, a change in the DNA. Sometimes the results of that are positive, sometimes the results of that are negative, but the mutation itself has no value until the environment in which the organism lives in decides whether that mutation and the resulting phenotype is beneficial to the organism. So the mutations are random, natural selection is not. So what do we mean by that? Well, these wildebeest all have these adaptations that helped them to migrate. And if those adaptations didn't occur, the species as a whole wouldn't survive. So they've had lots of random mutations within this population of wildebeest. But those mutations that provided a benefit for the organism to deal with this migration were kept and favored, and those individuals were more likely to survive, reproduce, and pass on those genes to the next generation. So we're going to move away from the wildebeest a little bit and look at another animal, the rock pocket mouse. And so this is taking place in the desert Southwest in the US. And if you'll notice here, and what you would expect, a desert would have light sandy substrate on the desert. Then we've got these little rock pocket mice, and they have a unique mutation. They have mutations that will affect their fur color. Some have darker fur and some have lighter fur. And if we stop right there, those genetic mutations have no value whatsoever, either good or bad. It's only when their environmental conditions, maybe even a disruption, selects which one might be favorable. So the population of the whole can change if that selection selects for certain mutations. So we're going to take it a couple of minutes of a little clip here, about 3 minutes, to go over these rock pocket mice.
Across the American Southwest, golden deserts dotted by cacti and brush stretch for miles. Yet here in New Mexico's Valley of Fire, the landscape changes dramatically. Patches of black rock interrupt the sand, remnants of volcanic eruptions that occurred about 1,000 years ago. The eruption spewed a river of lava more than 40 miles long across the desert. As the molten rock cooled, it darkened, leaving any creature dependent upon camouflage in serious trouble. In the complex battle of life, one of the constant struggles is between seeing and not being seen, the evolutionary game of hide and seek. And we've come here to the Valley of Fire in New Mexico, a battlefield, to find one of the tiniest soldiers and what it can teach us about how evolution works. On the desert sands, the rock pocket mouse blends in perfectly, its light-colored fur concealing it from predators. But on dark lava, the same fur makes the mouse stand out, attracting the many creatures that see it as food.
These mice are the Snickers bar of the desert. They're eaten by foxes and coyotes and rattlesnakes, and certainly by owls, and maybe even occasionally hawks. And most of those predators are visual predators. You know, we haven't been...
So what happened to the pocket mice that found themselves on this new terrain?
We did this big crossing.
When I accompany biologist Michael Nachman onto the lava, it doesn't take long to find out.
Oh, this one's closed.
Nachman has been collecting mice unharmed in traps. And it's a dark one.
It is, yeah.
Now, are most of the ones you find up here dark?
Almost all of them.
Not only have the mice here evolved to be as dark as the rock,
Shake them into the bag.
the color change has occurred precisely where it will conceal them from hunters. A bit of a white underbelly, too.
That's right. All of the dark ones here and on other lava flows have a white underbelly, and presumably there's no selection for dark on the belly because predators are coming from above.
Left to themselves, the mice show no preference for light or dark rocks. It's the predators that have made the difference.
The change in color over evolutionary time in the population is driven by predators weeding out the mice that don't match their background.
So pretty cool story and
But
a
how
great
did-
illustration of adaptation and evolution. So if we kind of look at a visual representation of what we just talked about, let's imagine this is a location where there's no volcano, there's no lava flow. And in this situation, the numbers of light and dark mice pretty much stay even. There's rarely a lot of dark mice because that particular mutation results in them being hunted more often. So when there's no volcano, population kind of stays the same. But if we look at a situation where we have a volcano, and then all of a sudden we can see the population start to change. The number of light mice decrease and the number of darker furred mice increased. So we can clearly see the effect of that disruption on the ecosystem. Now, one of the things you may be asked to do on an FRQ is to explain patterns and trends in data to draw conclusions. So we've seen this story in a video version. You've seen it in a little picture version on the last slide, but we could also talk about this in terms of a graph. So could you stare at this graph and tell the same conclusion? Now, what we can do is we can look at location A, which is the first two bars of each graph. And really location A really doesn't change too much in terms of the proportion of light and dark fur. But if we look at location B, we see the number of light furs decreasing and the number of dark furs increasing. So that is just another visual representation, quantifiable representation of the same story. All right, so let's look at one more example of this adaptation where an organism is evolving to adapt to a disturbance. Now, this particular one we're going to look at is an anthropogenic disturbance. It's adding antibiotics to bacteria. And so even though we're studying natural disruptions today, I wanted to point this out because this particular piece of content will come in handy when we think about antibiotic resistance within a CAFO, for example, or perhaps what's going on underlying the pesticide treadmill when we get to agriculture. So similar stories here. So this is a really fascinating experiment where they took a petri dish. So a petri dish is normally about this big, and they made a giant petri dish, a mega plate. And they grew some bacteria in there. But what they did is along the way, they would increase the amount of antibiotics. Now, antibiotics would typically kill the vast majority of the bacteria. But every once in a while, there was a mutation that allowed them to survive, and they flourished. So this is a great visual of that process. So here they're filling up the plate. And this is just the growth medium, what the bacteria grows on. Then they added in some antibiotics at differing concentrations. So if you notice here in this first section on the left and the right, there's no antibiotics. So we should see the bacteria population really thrive here as it grows toward the middle. But then here we should see a big stop because now we've added some antibiotics. So we should see the majority of the population stop right here. But what you'll see is they will evolve as they move towards the middle. And this is happening so quickly in 11 days because this species reproduces so quickly. Bacteria can reproduce in a very fast rate. So you're going to see a change in their population in much quicker time than you would in the mouse or even something like an elephant. That's going to take a lot longer to reproduce. All right, so increasing concentrations of antibiotics as we move to the middle. So you can see they stop at the border where the antibiotic starts, but those are the bacteria that have the adaptation for living in higher and higher and higher concentrations of the antibiotic. So if we stop here, this is the same species as this. It's the same organism. It is simply adapted and evolved over these 11 days to resist this level of antibiotic. Pretty cool. All right. So, excuse me, what you can see here is whether we're talking about mice or bacteria, or maybe even this scrub jay, which is a species under threat here in Florida, that our goal is to have a healthy, big population so that the genetic variation is maximized. We want the maximum number of genetic possibilities within a population. So that means maintaining a large, healthy habitat so that the populations don't get too small. If they get too small and the gene pool gets too small, they're more at risk of extinction. All right, so now we're going to go to the third topic of this unit, which is ecological succession. Still under the umbrella of change over time, but now we're going to look at how a single community changes over time. So to get there, we're going to define succession, and there's two types of succession we need to delineate, primary and secondary. Then we're going to look at pioneer species and their role in primary succession. That's a cool story. I like that. We'll look at the effects of succession. How does that change an ecosystem? Then we're going to look at two particular labels of species. One is a keystone species, and they have a very unique role in an ecosystem. And then we're going to look at an indicator species. That's something that we use in experimental work for a very particular purpose, so it has a utility for us. So that's where we're going. All right, so again, Ecosystems have structure and diversity that change over time. So if we look at this forest community, it is not static, it changes over time. If we look at this field down here of just little annual plants, over the course of decades, maybe hundreds of years, it becomes a forest. And that process is called succession. So succession is the sequence of changes in a community over time. Now, like I said before, we do delineate between primary and secondary succession. And really the distinction is quite simple. Primary succession happens when there's no soil. And that's a pretty rare event in terms of kind of wiping out the surface of the earth where there's no soil. Typically, we see it from things like the volcanic eruption we saw before. That would kind of wipe out all the soil. Or a glacier retreating, that would scrape away the soil. So when that area or that community of no soil is building up, that's primary succession. If there's soil already there, that's secondary succession. So if I'm interested in seeing how this community of perennial herbs and grasses is becoming a shade-tolerant tree-dominated forest, that's secondary succession. A good example of secondary succession is a forest fire. This is a longleaf pine forest from down here in the southeast, and we can see that this fire, which is natural, this is a natural disruption, has kind of cleaned out all the understory, the herbaceous grasses that existed on the understory, but they're starting to come back and then they reestablish themselves here. So this process of changing the community over time is called succession. So that makes sense. The soil's still there, so the plants can simply regrow back on the soil once the seeds or the spores land there. But what if we have a place where there is no soil, like that lava flow? So we've got a volcano, we've got some lava, it starts to ooze out everywhere and flow everywhere. And what we're left with, zero soil. Well, if an oak tree seed lands on that lava field, it cannot grow. There is no soil there. There's not enough nutrients or soil or anything for it to get going. So how do we even start this process? So before we go into the details, I do want to point to a great place to practice this. This is the 2014 FRQ about the Ring of Fire and plate tectonics and tsunamis. But part of that really lends itself to what we're talking about today. It says, although the landscape following a volcanic eruption may appear unable to support ecological communities, over time, the area can be transformed through succession. The first part says, what's primary succession? And the second part says, explain how primary succession can lead to soil formation. So exactly what we're going to talk about today. So primary succession, like we said, is the establishment of organisms or a new community where no soil previously existed. And B, or II, talking about how we get soil, that is where we see organisms weathering the rock and decomposing to add nutrients. So let's go into a little bit more detail about that process. So if we look at this early successional habitat, so this is one community changing over time, back here in the early stages, what kind of organisms might be most suited to live here? Clearly not a deer. Clearly not even an earthworm. There's no soil there for the earthworm to live in. There's no plants for the deer to eat. So what we're looking at are a group of organisms called pioneer species. And these pioneer species are things like lichen and bacteria and mosses, and they're the first ones to arrive. Now, this is a harsh landscape. There's not a lot of resources there. So these organisms have had to evolve particular reproductive and life strategies to allow them to live in these conditions. One aspect of their life is their reproductive strategies, which is in this case is an R species or an R selected species. And we'll talk about more R and K species when we get to populations. But these R species, one of their characteristics is they have a huge reproduction rate with low parental care. So if I'm a fern, I can distribute millions of spores and, peace out, spore, good luck. As opposed to a mother elephant that might care for her elephant for a long time. But anyway, that's a different story. What we're looking at today are these pioneer species, these ones that can kind of take this rugged environment. So one of the unique things that some of them can do, like the lichens, is secrete an acid that starts to weather down the rock. And when we get to soil, we'll talk a lot more about this, but soil is one of those beautiful places where the lithosphere meets the biosphere. It's like this recipe that is made from rocky stuff, and living stuff. Well, these lichens kind of get the recipe going. They break down the rock into smaller and smaller and smaller pieces, or at least get the process started. But that lichen or that moss or that fern is eventually going to die. And when they die, they decompose and their organic matter gets mixed in with that weathered rock. Boom, now we've got some soil. It's not a lot of soil, can't grow an oak tree yet, but we've got a little tiny bit of soil, broken up rock with some decomposed organic material. That now can maybe support a little larger plant. And then that plant dies, and maybe it supports a larger plant and maybe a spider. Well, when those things die, so you can see that over time, the soil gets bigger and bigger and bigger, and we can support more and more life. All right, so here's a picture of this. So we start over here with just bare rock, and over time, we get to this forest community that has a really biodiverse, species-rich ecosystem. So the succession of plant communities above the ground is kind of mirrored by the soil development below the ground. We can see the soil going from non-existent to all of these horizons, a very rich and developed, mature soil profile. So over time, we can see changes in biomass. Clearly, there's much more living stuff in late successional communities. Species richness, vastly different from what we saw here in primary succession. And then NPP, think about that. Think about the difference between NPP of a bare rocky area, almost like a tundra or a desert, versus a tropical rainforest or something that's here. So I'm not saying that deserts become tropical rainforests, but I'm saying over time, as a community goes through succession, its NPP changes, its productivity changes. So I want you to imagine now, what if I took away this fern? What if I took away all the ferns from that community, that late succession community? Would it affect the community? Absolutely. The things that ate the ferns might decrease. Maybe the nutrients in the soil might shift because the ferns aren't drawing that out. But by removing the ferns, or if I remove the mold, or if I remove the fungi, the forest is still staying a forest. However, there are some organisms that, if they are removed, completely changes the successional pathway of a community, and that community can become a completely different one. So what I'm saying here is not all species are equal in terms of how a community functions. Some you can take out and it might shift a food web or change a food web or soil dynamics, or it might change something, but there's others that if you take it away, the whole ecosystem changes, the whole community changes. And that's what we're going to look at through the famous work of Dr. Robert Payne, brilliant scientist who was really famous for this first experiment he did with starfish, talking about the role of keystone species. So he investigated tide pool communities, and he studied how the removal of this one species of large starfish would affect the rest of the community in the tide pool. Again, an extremely important experiment in ecology. So he was really interested to see how these kind of apex predators like these starfish regulated the biodiversity of an ecosystem. So one way to do that is to kind of make an experiment where he removed some, and you'll see how he did that here in this clip. We're going to watch just 2 or 3 minutes of this.
He began by identifying all the organisms, and then he started mapping out who eats whom.
There were carnivorous gastropods feeding on barnacles. There were sea urchins feeding on algae. There was a lot of pattern.
His observations showed that a species of large purple and orange starfish, called Pisaster ocraceus, was at the top of the food chain. Starfish may seem like unlikely predators, but speed time up a bit and you'll see deadly hunters.
If a starfish is feeding, you turn over and you see what the starfish is eating. They're eating mussels. They're eating a lot of other things as well, but they're eating mussels.
So Payne asked, what happens when you remove the predator starfish from a single outcrop?
You have to surprise them because the starfish clamps down. It takes a strong wrist and a pry bar. I would then scale them as far as I could, and in those days I could throw a starfish 60 or 70 feet out into deeper water. There were always starfish marching in, so during the summer months, I would drive the 350 miles round trip, hit the area at low tide, do my removal, take other data, and then return to Seattle.
The ecosystem started to change rapidly.
Within a year and a half, I knew that I hit ecological gold.
Although the top predator had been removed, surprisingly, the number of species in the rock actually decreased.
So that's a little different than what we would expect. You would think if we took away this top predator, that the other species below would be like, oh, thank goodness the predator's gone, and the biodiversity would increase. He saw exactly the opposite, and here's why.
From 15 to 8.
After 3 years, it went down to 7, but then by another 7 years, it had simplified itself till it was basically a monoculture. I changed the nature of the system.
As the experiment continued, the line of mussels advanced down the rock face, monopolizing almost all of the available space and pushing all other species out. Payne had discovered that one predator could regulate the composition of an entire community. He coined a term to describe the power a single species can exert on an ecosystem.
I know very little about architecture, but if you build an arch, you have to get the two sides of it to put pressure against one another, and therefore at the apex of the arch, you have a keystone. You pull the keystone out and the structure collapses.
So very cool study that he did. So again, Thomas Payne's work, or Robert Payne's work on keystone species is very, very important in understanding succession. So this species has a crucial role in maintaining the community structure. It affects lots of other species in that community, can really drastically reduce the biodiversity of an area. This is a unique characteristic of a keystone. Typically, it's not the most abundant species. Therefore, the impact it has on the community is much greater than you would think because of its low abundance. But its absence would affect the entire structure or functioning of the community, and it kind of alters that successional pathway. So we see here that early, before he went and removed the starfish, we had lots of biodiversity. After removing the starfish for a year, it started to go down. 5 years later, the species richness was reduced to 1. So we saw this one organism not just affecting the things that it eats and what is eaten by it, but the entire rest of the trophic levels. And we call that effect a trophic cascade because its effects cascade down to other parts of the food web. So another good example of that is an African elephant. So one of the things that an African elephant will do as it eats vegetation is strip trees down, knock trees over, trample them, and it really reduces the amount of forest. This actually keeps the African savanna a savanna. If you removed the elephants, then the ecosystem would actually go through a different successional pathway and become a forest. So you can see that this one species, not a very abundant species, has the ability to shape the entire structure or function of a community. Another famous example is with wolves. So over here on the left-hand side, this is before the wolves were removed. So we have a large mix of small and large predators. There's a diverse plant community, lots of birds, few large herbivores. But once those top carnivores are removed, the smaller predators dominate. And so what we have is a big explosion of these herbivores, which reduce the plant community, the plant biomass, and therefore reduce some of the songbird diversity. So again, one organism, the wolf, having this cascade effect all throughout the community, this trophic cascade. All right, so the last type of organism we're going to look at is an indicator species. And this is a species that we define as a useful way to count and understand the health and stability of an ecosystem. So let's take this wood stork over here at the top right. This is a common bird in the Everglades or in kind of marshy wetlands here in Florida. And so one of its habitat requirements is, in order to feed, it needs shallow, rather turbid, seasonal pools of water in which fish are concentrated. And what the bird does, it'll open its beak and kind of wade its beak through the water with the beak open. And as soon as it feels a fish, it'll slam shut. Well, that ability, that adaptation, can only occur in shallow, turbid, seasonal pools where fish congregate. And that's a natural state of a marshland like the Everglades. So therefore, if I go out to the Everglades and I see lots of wood storks, that's an indicator that the ecosystem is doing well. But if I go out there one season and the numbers of the numbers of wood storks have gone down dramatically, that means that something is up and I need to look further. It indicates something is amiss. Now, I picked the wood stork as my indicator species because it has a very specific habitat requirement that really defines the ecosystem. Here's an example of what you would not pick: a raccoon. A raccoon could easily live in that salt marsh, but it could easily live in my neighborhood by eating trash out of the trash can. So that kind of generalist species that isn't really connected intimately to one ecosystem would not be a good species to call out for indicator. And then down here at the bottom, lichen. Lichen is a famous indicator species for air quality. So overall, an organism whose presence, absence, or abundance reflects a specific environmental condition. In this case, shallow, turbid seasonal pools. In this case, good air quality. And again, it gives us an overall indication of the health or the equilibrium of an ecosystem. And it's usually an organism that you could count or quantify pretty easily. You wouldn't want something that is almost hard to detect. Finally, a nice one to think about is amphibians, such as frogs. Because of their intimate relationship with aquatic environments, they're a great indicator species for water quality. That's often used. All right, so let's do a quick check for understanding. Again, I'm going to give you some quantitative data in the form of a graph to understand and ask you a particular question. All right, so the question says the data on the left represents the normal biodiversity of a community. The extinction of the crab, a keystone species in this community, would most likely result in which of the following? So take 30 seconds for that question. So the answer here would be an increase in the algae biomass, because the removal of this crab would cause a population explosion in this fish, which would cause a very low population of these organisms, and therefore the algae would increase. So this is a good example of a trophic cascade, where the abundance or absence of one organism has a very big effect on the rest of the community. All right, so where did we go today? We looked at disruptions, natural disruptions, adaptations, how organisms evolve to adapt to those changes. And then we saw ecological succession, how a community itself changes over time from primary to secondary succession. And then how certain species like the keystone and indicator species are useful for us to understand those areas. Just as an aside, to think about conservation strategies, if you only had $5 to spend to save one species, maybe you save the keystone species, because that does have such a large influence on the ecosystem. So what am I going to leave you with tonight? Another FRQ for practice. So again, pay attention to task verbs, and this is the question that you're going to be answering for homework, and Ms. Fassler will talk about this tomorrow. This starts out with content that we have not got to yet in the review: generation of electricity at a hydroelectric dam. But you've covered that content in class before, hopefully, so that part will be a review. But towards the end, it talks about keystone species and the roles beavers play in those ecosystems. So this is the FRQ I will leave you for next time. And again, please reach out if you have any problems with connectivity or the mobile tools that you're going to need for this course or for the exam coming up, or if you know anybody else who might, please use that URL below. And again, thank you guys so much for doing this and sitting here and listening to me ramble on and on. Ms. Fassler and I are so thrilled that we could be doing this for you guys. Hopefully it's beneficial to you. And I'll see you next time. Bye, apes.